So in the lecture today, we will now move to mutation and transformation. Last time we are still at the cellular level. So of course we will see later cancer as a stochastic effect of radiation in humans. But at the moment we stay in this lecture at the cellular level and we see mutations and neoplastic transformation, which is, let's say, the cellular equivalent of cancer in organisms. So mutations, of course, mutations is, for radiation-induced mutations is something very well known. Is in our imagination, is especially in terms of hereditary effects, is actually not really like that. We will see that hereditary effects of radiation are very seldom. But mutations are popular in the literature. You see Spider-Man, Fantastic Four, Hulk, Godzilla. These are all reported examples in the comics books of mutations, somatic mutations induced by radiation that give superpowers to the person that are affected. This we will see never happens in the real world. I mean, you don't get superpower, but you get cancer. So it's not good. They're not good. But this is the general how popular is the topic of mutations. In fact, let's start. So now we will start to speak in general about mutations. What is a genetic mutation? A mutation is always a change in the genetic material. And of course, a mutated gene can produce a flawed protein that does not function properly or does not function at all. There are many examples of genetic disease that are related to the change in one particular gene. Typically, these are hemophilia, sickle cell anemia, and cystic fibrosis, but there are many, many others. These are examples of the blood cells in sickle cell anemia. The causes of mutations, of course, there are many, many causes of mutation. Viruses induce mutation, UV radiation induce mutation, chemicals, pollution, all these environmental factors induce mutation, and of course, for us, ionizing radiation. Now, there are the gene mutations can be classified in different ways. The base pair substitution, also called point mutations, are maybe the most easy to imagine, because in that case you have, for example, here you essentially change. You make one change in one specific base. In this case, you see the C, the cytosine that is read as guanine on the messenger RNA gives rise for this codon to an arginine. But if you replace the cytosine with thymidine, at the end, the codon in the messenger RNA is CAU, and this is histidine, is not anymore arginine. This point mutation, so you change only one base, but then you have a different amino acid, so you have a different protein. That's enough to induce most of the genetic disease that we know, like cystic fibrosis, type B blood, sickle cell anemia, and so on and so forth. However, as you can easily imagine, there is quite a number of point mutations that are silent. These are called silent mutations, so it's essentially a change in a base pair into another that codes for the same amino acid, because the code is redundant, so you can change it the same. And an example is the type B blood. These are mutations that don't really change the amino acid sequence. The missense mutations instead are point mutation that code for an amino acid, but the wrong amino acid. This is what we have seen previously. The typical point mutation is a missense mutation. So now you change amino acid. So the mutation causes a change in the amino acid. The nonsense mutation instead, essentially because of the point mutation, this is another type of point mutation, because of the point mutation now, you don't code for an amino acid, but you code for a stop codon. So while you are quietly building your protein, now instead of finding an amino acid, you find a stop codon. So the protein will be truncated. And insertion instead is one or more base pairs are inserted into the code. Of course, this causes a shift in the reading frame, generally. And the shift in the reading frame is, of course, a big problem because now everything will be read incorrectly. So the result is generally missense mutation or nonsense mutation, depending on where you have the insertion. And finally, deletion. Deletion is the simplest type of mutation. It's also a very simple type of mutation. Here, you essentially delete, you lose one base. This also causes a shift in the reading frame, and then cause missense or nonsense mutation. Actually, we will see that most radiation-induced mutations are deletions, or they derive from chromosomal rearrangements. Of course, a chromosomal aberration brings mutations typically with it. So deletions are very important for chromosomal aberration, for ionizing radiation. For UV, for example, point mutation, the thymidine dimer is more common, but for ionizing radiation is more common the deletion, because most of the damage, you know, the irreparable damage is done at the strand level. In fact, let's go to the radiation-induced mutations. This is a very old story. It starts in 1927 when Hermann Müller actually irradiated mature fruit flies. Fruit flies, Drosophila, you know, is like the basis of genetics. It's like the pendulum for the physicist, for the genetist, it's the Drosophila. He irradiated with the fruit flies with radiation, measured the lethal mutations. And he used very high doses, you see here, and he got a linear dose response. So this is actually an experiment as old as 1927, but it's still the basis of how we build radiation protection. Now, radiation protection is divided, we will see this, is divided, of course, in radiation protection for acute effects that are related to high doses, and for stochastic effects. Those for stochastic effects is related to mutation. So we want to prevent the possibility of mutation. The work of Hermann Müller, that got the Nobel Prize for this, inspired the National Council for Radiological Protection in the United States to lower the radiation limits that before were related to skin erythema, essentially to acute effects, already in 1932, and to propose the linear no-threshold model. Linear no-threshold means that the probability of mutations is never zero. It's zero only for zero dose. So it's linear and there is no threshold. Why this no threshold is so strange in toxicology? Mutations are studied in toxicology. The idea of having a linear no threshold model in toxicology is very strange. We will see why. But for radiation, it was adopted based on this graph, on the results of Müller, and is still applied. Still in radiation protection, we use the linear no threshold model. Not without discussion, we will see, but it's very unlikely that we will change. And we can argue why, but it's very unlikely that this will be changed. One very nice way to measure the mutation rate in cells is to use the so-called HPRT assay. The HPRT assay is based on the hypophosphorybosyl transferase gene, which is a particular gene located on the X chromosome, to be precise, in the XQ26.1 band. If you do the G bending. And why it's so important? Because the wild type, so if you have no mutation in this gene, the cells will not survive in a nucleoside analog 6-thioguanine. So essentially, if you if the gene is not mutated and you grow the cells in the toxic nucleoside analog 6-thioguanine, they will go into S phase using this nucleoside, and then they will die. So it's lethal. So normally they die in the presence of 6-thioguanine. But if you mutate this gene, they will not use the 6-thioguanine nucleotide, and then they will survive. So it's a very nice assay because you look at the survivors, and from the survivors are the mutants. So it's not a negative assay, it's a positive assay where you see the mutants. Mutations are very rare, we will see. So having this kind of assay is very convenient. So these are, for example, old experiments from the 20th century, let's say, on the induction of mutations in the HPRT gene as a function of the dose. These are mutations for 10 to the 6 irradiated cells, here and here. This is with X-rays, and you see the linear quadratic dose response typically. And this is with alpha particles. Okay, this is not as a function of the dose. Dose is here, I'm sorry. Here is the fluence, here is the dose. Of course, dose and fluence are the same thing. And you see the linear behavior. So again, if you remember, let's say the microdosimetry approach, linear quadratic response when you have X-rays, purely linear response when the intra-track events are much more likely than the inter-track event, and then you have linear response where the beta term goes to 0. This is a more recent review and calculation of the mutations in the Chinese hamster ovary cells in the HPR3 gene. You see, it's a collection of different experiments that show the average mutation rate per 10 to the 4 cells. Okay, so you see linear quadratic, nicely linear quadratic, is measured in different experiments. But this experiment here, he also measured point mutations. And you see the point mutations are much lower. And also they are not linear quadratic. They are essentially linear. And this makes sense if you think about it, because the linear quadratic behavior here shows that these mutations are related to chromosomal aberrations. But the linear behavior here means that you can induce point mutations with essentially are all intra-track events. It's just a mutation induced by a single track, so it must be linear. But you see that it is much lower. It is much lower. You see, it's less than 10% of the total mutations are point, approximately 10%. I mean, approximately 10% of the total mutations are point mutations. So it's a relatively low level of mutations are really point mutations. If instead, you see, these are all experiments with X-rays, but if you do the experiment with a chemical, like MMS is a toxic chemical, is a mutagen, a very well-known mutagen, and this is now the dose, but the dose, of course, is not in gray. The dose now is in microgram per microliter, so it's how much chemical you put in your dish. And this is actually the mutation per 10 to minus 6. So you see, it's quite higher than this one. If you look, I mean, these are per 10 to the 4, 1.2 per 10 to the 4. This is 10. Yeah, it's higher, I mean, but not so much higher. This is like 25 per 10 to minus 4. But what is very important here is that you see, there is a threshold. So it is the linear no threshold that we use in radiation is generally not found in toxicology. So when you add the poison, you normally have a threshold. You have a level where you have no effect, and then you start to have an effect. This is fairly common in toxicology, and is the reason why there are threshold levels in different chemicals that you have in the water or in the food. You know, there is a minimum level. This makes sense because you can assume that below this minimum level there is no effect. But for radiation, you will not use this kind of, let's say, the community does not use this kind of dose response curve. As a matter of fact, let's try to compare chemicals and radiation in terms of the mutations. This is an example, V79 Chinese hamster cells. This is a typical cell-killing, cell survival experiment, and this is now a typical mutation induction experiment. Now, this is not necessarily linear. There can be a curvature here, but there are only a few points. For our argument, that's good enough. I mean, you don't see the curvature that could be here. It looks like linear, but it's not necessarily linear. And at the same time, you see, this is now EMS. EMS is the ethyl methansulfonate, while the MMS is the methyl methansulfonate. This is a very powerful mutagen, but it's also a killer. You see, this is the survival curve. Now, this is not the dose in sievert, but it's a dose in concentration in micromolar. This is the micromolarity of the ethyl methansulfonate. And this is the mutations where, again, you don't see, there should be more like a threshold here, let's say, but because there are only a few points, you fit it now linearly here, but it's more likely that you have a behavior with a threshold and then linear. But what is the point? The reason why I wanted you to compare this one is that now I can calculate for radiation and for the chemical, the mutants per survival. So now I have the scale of the dose here, so I can calculate the mutants per survival. And that's how it looks like, you see. For EMS, these are the mutation, and this is the percent of surviving cells. For UV, you have this curve, but for a relatively poor mutagen like MMS, or for X-rays, you are here, where you see you are very good in killing, but you are not so good in inducing mutations, where you see EMS is very good, is much better in inducing mutations than in killing, and even UV. So generally speaking, X-ray ionizing radiation is a very good killer because it causes macroscopic damage in the cell, while many not all of them, but many chemicals are very good mutants because they can induce mutations without killing the cells. But generally speaking, X-rays is a poor mutagen. And it's so poor that you wonder whether it makes sense at all. You see, this is it's a controversial plot, if you want. It gives you the number, it goes down to the mutation, and the number that you see in parentheses is the fraction of metabolic DNA damage per 1 millisievert radiation in 1 year. So let's take a typical environmental exposure, 1 millisievert radiation in 1 year. And let's see what is the metabolic DNA damage. So spontaneously, so independently of radiation, what is the fraction of free radical and reactive oxygen species that you have in 1 year? So you will have 10 to the 8 free radicals and reactive oxygen species produced, but this become 10 to the 6 if you look at DNA oxidative adducts. So there is a factor of 100 if you go to the DNA oxidation, the attack. But this is only 5 per 10 to minus 9 of the spontaneous. Then this again goes down to 10 to the 2 if you look at misrepaired or unrepaired DNA alterations, 10 to minus 7. And this corresponds to approximately 1 mutation if you go to, and this still 10 to minus 7. So you see, of course, what happens here is the prevention. You can have antioxidants that reduce this passage, repair, reduce this passage, and removal, for example, apoptosis, necrosis, is reducing this last passage here. So 10 to minus 7 is an unbelievably low number. So you wonder, why do we care about 1 millisievert of radiation? Well, apparently so. But on the other hand, the damage which is induced within the cell spontaneously is not really the same as the damage induced by radiation. This we have seen. Even electrons can induce clustered damage. So this passage, let's say, in repair and removal is not necessarily the same. So the damage induced by radiation is sometimes unique, of a unique type that is not reproduced in the environmental, in the spontaneous errors of the cells in replication. Now, finally, the RBE. How is the RBE as a function of LET? No surprise here. This is a collection of data for HPRT in CHO cells. Actually, this experiment was done at GSI with many different ions. The points are all the experimental data with different ions. So you see that they are not really, there is a lot of scatter, but the behavior is essentially always the same. This curve here is the current ICRP, so the International Commission on Radiological Protection curve for the RBE as a function of LET. You see, it's not exactly the same. Looks like here, for example, goes down more than the ICRP. But if you take mutation, RBE versus LET, you are not very far from what the radiation protection is using. So this kind of bell-shaped curves, we have seen them already. We have seen them with chromosomal aberrations. We have seen them with cell killing. Now we see them with mutations. Okay, but what are the consequences of mutations? The most worrying consequence of mutation is, of course, is neoplastic transformation. What is neoplastic transformation? Neoplastic transformation is in vitro what we will then see in vivo. So it's called the neoplastic transformation, what we study in system, in the lab, but in the humans will be called cancer. And we have already seen that is a multi-step event. We have discussed this in detail in the biology. So please go back to the biology lecture here, because we have seen that the model of addition of mutations, you have a series of mutations, like 7, 8 mutations, and then eventually becomes a cancer. This is kind of challenged recently by more studies that show that the microenvironment is probably much more important than the mutation events. But let's stay on this model for now. Let's see what we can do in vitro. Just one word on in vivo. We will come back to this when we do stochastic. We know that radiation induced cancer. We know this for sure. The for sure comes mostly from Hiroshima and Nagasaki, because in Hiroshima and Nagasaki, we had a population of people with the same genetic strain. They were all Japanese, that were simultaneously exposed to different doses of radiation. So we built nice dose response curves. And we know also that the latency time from the exposure to the cancer is very long, which is something that you can never reproduce in vitro. Leukemia is relatively small. There was a peak of leukemia within 5 years from the bomb, and then this one decreased. There is a long latency of something like 10, 20 years for solid cancers. And there is now, is clear, also a long latency for other disease, like heart disease. We will see this in the stochastic effect lecture. We have also other cohorts. A very famous one is Chernobyl, but as we will see in the radioecology lecture, Chernobyl was a special case. Most of the cases in Chernobyl are related to the thyroid cancer. You see a very nice dose response curve here from Chernobyl, but this was mostly due to the fact that we will see this in Chernobyl, that the population was not alerted of the nuclear accident. The iodine could be deposited. It was eaten by the cows and then ended up in the milk, and the population was still drinking fresh milk, especially the babies, the children. And this is mostly pediatric cases. And then there is a number of other cohorts. You know, the radium dial painters is very famous because they were the person that were leaking the paint on the watches, and there was radium there. Thorium injection. But if you see the real number are relatively low. So if you look at the excess cancer death case, they are always very low, even Hiroshima and Nagasaki. Now it's not, this slide, I apologize, I have to update it. It's not anymore 428 because there are still people dying in Hiroshima and Nagasaki, of course, because there's still someone alive. So it's higher, but not much higher than that. I think it's 600, 700 now. And you see also the other numbers are relatively low. And there is a large excess of childhood thyroid cancer in Chernobyl. We see something like 1,500. But these are not lethal cancers. Well, these are really lethal cancers, but these are not really lethal cancers. So epidemiologically, the cohorts where we study radiation effects for cancer are extremely low. Because radiation, as I said, is not the powerful mutagen and is not the powerful cancerogen. So it's the impact, if you remember also the curve where we saw how important is the number of stem cell divisions in the risk of cancer in the different tissues, and you have and what is the impact on the environment on top. If you remember for lung, for example, smoking was a factor of 10, an increase of a factor of 10. Radiation is never like that. Is impossible. It's always a very small increase. And that's why in vitro studies or in vivo studies are important, because we can try to study. It's very difficult to get all the data from epidemiology. It's more easy some data, some results, you can get them from lab studies. In vitro, the characteristic of the cancer cells in vitro are quite easy. The normal cells grow and they reach the contact inhibition. So when they have completely filled the flask, they stop. You see, if you remove some, they grow again and then they stop again. But the tumors don't stop. They grow on top of each other. So the clones done by the tumors are quite easy to see because they are growing also in the z-axis, if you want. And there are also other systems to see them that are really, really interesting. One case is the HeLa per skin fibroblast system. So what do you do here? Here you fuse HeLa cells, which is a cancer cell, with skin fibroblasts, which are normal cells. Now, generally speaking, this was an old experiment already done in the 70s. If you fuse a cancer cell with a normal cell, generally you get a normal cell. This is interesting. And the reason, again, is that the tumor suppressor genes in the normal cells can now suppress the expression of the oncogenes in the cancer cells. So the fusion normal cancer makes a normal cell. So these cells, CGL1, they are called CGL1, they are normal. However, they are, of course, prone to transformation. And they are particularly nice because they, once they are transformed, so once they go into the cancer stage, they can be easily stained, so they can be easily seen in the dish. Rodent cells can also be some kind of predator. So these cells, I insist, they are not really normal. The C3H21 half cells that are mouse embryo fibroblasts, they are really, if you look at the karyotype, they are completely aneuploid. They are not normal at all. And this is a system where if you irradiate these cells, you get a lot of colonies like this. You see them in the dish already. Colonies transforming colonies. So if you count the number of transformant colonies, you can get the frequency of transformation. But at the moment, in the lab, not a single normal, really normal human cells have ever been transformed in vitro. That's to tell you that it's not easy. You know, so in vitro, if you take a completely normal cell, you don't get the neoplastic transformation stage. You must use something which is pre-transformed. I mean, some kind of already quite ahead in the stepwise cancer induction. And that's how the dose response curve will look like. So you have to think to what, if you calculate the number of transformants per cell exposed, you generally get a curve which is like that. Why is that? Because you have an increase if you look at the transformants per surviving cells as a function of the dose, but then you have the cell killing, which is exponential. This curve here is generally linear quadratic. So this is actually a log scale here. So this curve is linear quadratic. This curve is exponential. So if you look at transformant per cell at risk, it goes down. So make no mistake, because sometimes you will see transformants per surviving cells, sometimes you will see transformants per exposed cells. And the dose response curve is, of course, very different because it depends on the impact of the cell killing. Now, if you go to low dose, this is quite controversial, because this is a number of low dose experiments also done in C3H21 cells, in these CGL1 cells, and some data from Hiroshima and Nagasaki are here. This is the risk, so the ratio, if you want, this should be 1 if there is no difference. And you see, and this of course goes up at high doses, but at low doses seems to be even down, which is not only the threshold model that we have seen in toxicology, but even a sort of hormetic model. Hormesis is also known in toxicology. This means that if you take low dose of a poison, you become resistant. It's very controversial in radiation biology. The controversy is, let's say, somehow fueled by the high background radiation areas, that are the areas this we have seen in the first lesson, when you have very high radiation dose, but you have no evidence of carcinogenesis. It's not completely solved by Hiroshima and Nagasaki. These are different attempts to fit the low dose region of Hiroshima and Nagasaki data. And you see that it's compatible with the sublinear behavior here, but it's also compatible with the supralinear behavior here. And it's not fixed. You know, epidemiology can never fix this problem. You see, this is radon, lung cancer in radon. Again, you don't really can fix it because the variability is too high. So that's why we can think of using in vitro system. These are actually data on the neoplastic transformation of this human hybrid cell CGL1. This is the number of cell, the transformation frequency per cell at risk. So in fact, you see the decrease with the X-rays. These are 15 MeV X-rays. High energy carbon ions. Then when you increase the LET, you see you have more transformation, but you also have a higher decrease. This is the data plotted as a function, transformation versus surviving fraction. So you see that when you go to low surviving fraction, you start to see a decrease. Now, if you calculate the RBE now, the RBE for neoplastic transformation, and you plot it as a function of the LET, this is done in these are 3 main experiments, different LET with C3H21 mouse cells. And these are 2 experiments with Syrian hamster embryo cells. Again, something that is very familiar to you. You see the general trend is 1 at low LET, then you go up around 100 kb per micron, and then you go down again. This is the RBE for transformants per surviving cells. So it's corrected for the survival. Now, if you go to animal, of course, in that case, you cannot correct for survival. So if you look to the incidence of leukemia here, it goes up, then maximum, and then goes down. But you have to be very careful with the animals, though, because, you know, the problem that you have with the animal studies is that, in principle, you can get any kind of results depending on the susceptibility of the system that you are using. This is an example. The leukemia, acute myeloid leukemia, in the black mouse, for example, if you irradiate the black mouse with 1 gray of radiation, the incidence of leukemia is less than 1%. But if you irradiate a CBA mouse or C3H mouse or RFM mouse, you see here Well, at 100, this is in rad, the dose is not in gray, of course. With 100 centigrade, you see it's around 20%. Here is written 25% if you look at this animal, so very high. If you look at the F1, so if you mate the C3H mouse or the CBA mouse with a black mouse, with a black mouse, and you get the first generation, then you have an intermediate frequency of 7%. So the genetic component is extremely important. This has been always considered a drawback, but we will see some cases when we study stochastic effects and also the modification of radiation damage. We're not really sure that this is not also the same in humans, you know, that you can have exactly the same in humans. So what if some of us are black mouse and some of us are C3H? Can this explain the incidence of leukemia, the dose-response curve for leukemia, only because some individuals are very resistant, a majority, for example, is very resistant, and only a few of them are very sensitive? So this is a very interesting topic, as you can imagine, really fascinating topic. I will show you some real data. And these are also some, to show you how complicated is the story. You remember that I told you that the image of the model of mutation, accumulation of mutations, does not explain the whole story, if you want. This is an example. You take a mouse, you clean the mammary gland of endogenous epithelium, then you irradiate whole body, then you transplant a TRP53 null, so is a cells that have no P53, and then you look for cancer. Now, what happens is that, which is really interesting, if this is the dose, this is the glands with tumor, and this is tumor-free as a function of the days. Essentially, you see, when you irradiate, you have an increase in the tumors, and even more interesting, you have a reduction of the latency time. So irradiating the host, not the tumor, but the host, reduce the latency. So again, this can be explained as a sort of inflammation effect, you know, some promotion that comes from inflammation. And that's the case also here. When you irradiate, this is a nude mouse, so it's not an immunocompetent mouse, is a immune deficient mouse. But you see, if you irradiate with gamma rays, this is the frequency of murine astrocytes that you get. This is what you get if you irradiate with iron ions, high LET. You see the frequency at the end is the volume is the same, but this occurs earlier than here. So it's not only the frequency that matters, but also the latency time. Of course, this is very true for us. I mean, if we take something and we are told that the latency time is 50, if you tell me that if I do something, I will get cancer, but in 50 years, I don't care, because in 50 years, I will be dead anyway. So I don't care about the latency time, you know. I don't care about, I mean, the latency time is extremely important. So one other story, they tell me that if I do something, I get the cancer in 1 year. Then I don't want to do it. But if they tell me in 50 years, who cares? I mean, I don't care. You care, because you would still be alive, but I will be dead. So there is no much discussion. These are other examples that show you how the story of the RBE, when you go to animals, is kind of complicated. This is an experiment, again, of induction of acute myeloid leukemia in mice. These are sensitive mice, you know, the CBA and the RFM. This is with gamma rays. This is with neutrons. You see, neutrons are much more effective, apparently. So they did the experiment in 2009. They did the experiment with iron ions, 147 kV per micron, the peak of effectiveness. They were expecting a higher B. The white points here is iron ions. The black points are gamma rays. No difference. RB 1. You see, 1 for iron ions, about 10 for neutrons. So, and also, you know, in the same system where they found no leukemia, they found a lot of hepatocellular carcinoma. The RBE for hepatocellular carcinoma is something like 20. Very strange, no? But if you think about, this is related to the problem of cell killing. So leukemia is a typical aberration caused by specific chromosomal aberration, a typical cancer caused by specific chromosomal aberration. It's in the mouse, it's also well known, it's a deletion. I mean, in the human, it's the Philadelphia chromosome. Now, if you kill the cell, if you are in a situation where you have an over-dispersion in the aberration, in the distribution of the aberration, where given a dose, you have many cells with many aberrations, including lethal aberrations, those cells with the mutagenic aberration will be lost somehow. So even if you induce more aberrations, you lose them because you have an over-dispersed distribution. So that's why the RB is 1 for AML, which is a cancer strongly related to chromosomal aberration because it's a liquid cancer, but it's very high for hepatocellular carcinoma that instead is a solid tumor, very much related to inflammation. So in the liver, you know, the liver cancer is typically caused by inflammatory agents, you know, by alcohol, for example, cirrhosis, by the virus, by HPV. So it's a typical inflammation tumor. And this other example, this is data where you see the RB for Harderian gland tumors in mice. This is very much studies. You see a very nice RB. This is the fluence, you know, going from protons, helium, neon, iron. You see that you have a much higher here. But keep in mind that Harderian gland is a gland in the eye of the tumor, does not even exist in humans, and is very much prone to transformation. And these are mammary tumors in rats, iron ions, protons. And look here, you see that for photons, looks like there could be some kind of hormesis here. It's not very clear, but there could be something like that. Okay. But in the model that we are working now, we always have the idea, in any case, that we have mutation, transformation. We already challenged this in biology. Let's see, from radiation biology, there are at least 2 effects that challenge this image, the so-called non-targeted effects. So non-targeted effects are divided in two main regions. One is radiation-induced bystander effects, which means that what can happen is that you hit the cell, but other cells are involved. And the other one is the so-called genomic instability. This means that you irradiate your cells, and then you wait, and then you start to have instability events that eventually lead to all kinds of events, from cell death to transformation and so on. So in genomic instability, you see the basic idea is that misrepair DNA damage, like translocations, mutations, is transmitted to all viable daughter cells. So you irradiate one cell. If this mutation or chromosomal aberration, whatever, is non-lethal, this will be transmitted to the daughter cells, and all the daughter cells will bring this aberration. Now, if you look at the idea of genomic instability instead, is that radiation exposure, you have misrepair, you have damage, whatever, destabilize the genome. So the genome is kind of becomes unstable. You cannot see it for a long time, but then suddenly you start to see effects like gene mutations, chromosomal aberrations, and so forth. This is more similar if you think about what we see in vivo, where essentially there is a long latency time in most tumors, not tumors like leukemia where the latency time is short because it's a liquid tumor, so it's chromosomal aberration involved. But for solid tumors, you have a long latency time where probably some promotion effects happens to transform this initial destabilization in genomic instability. And this is, of course, clear in carcinogenesis. You see, where you have genomic instability observed in cells with genetic disorders that are susceptible to cancer, and also aneuploid multiple chromosome changes that are found in cancer cells. Cancer cells are extremely genomically unstable. Sometimes it's very difficult to have two cells with the same karyotype in the same tumor, and they are very aneuploid. So the number of cells is, the number of chromosomes per cell is very variable, and multiple chromosomal changes. So genomic instability is a genome-wide process that can be induced by radiation, both high LT and low LT. At the beginning, high LT seems to be more effective, but now it's unclear. Epigenetic factors may significantly influence the development of genetic instability in vivo, that's for sure. Instability is manifested in different endpoints, but we don't really very well know the relationship. And the mechanisms leading to instability are complex. This can be deletion of genes that control stability, induction of genes stimulating instability. You know, it's something that comes with the time. Bystander effect is something that was seen in a very funny experiment. It's very much in vitro at the beginning. You know, one thing was, for example, you irradiate your cells, then you take the medium, not the cells, but the medium, and you transfer the medium to normal cells, and these cells die, not all of them, but some cells die. Or if you want, you irradiate cells here, and you put another well with other cells here, then you irradiate from the bottom, then you remove this one, and you take these cells in another plate, and then you find out that doing this, let's say, you can increase or reduce the bystander effect. Depends. Let's say if you put the insert after the irradiation, then you have more cell death. If you remove the insert after the irradiation, you have less cell death that you would expect. Or also, you know, there are other funny approach, you know, like in this case where you can irradiate the medium only, you don't irradiate the cells, or here you irradiate the cells and then you look at the depth of these cells here. So these are funny experiments, but if you think about it, they are all very far from in vivo. And also from understanding very well the mechanism. The best way is to use the microbeams. The microbeams are special accelerator devices where you can actually irradiate cell by cell. You see, you have a beam which is generally low energy beam because you have to focus it in the micrometer, sub-micrometer region. Then you have a microscope, you look at your target, and then you open the shutter and you shoot a given number of particles that you can count with the scintillator, with the particle detector of the microscope. So you can do funny things like this. You see, this is done at GSI. This is a human fibroblast, and we wanted to write GSI on the cell. And you see that shooting with the microbeam according to this pattern, you get a pretty good result. You see GSI. This is now stained for gamma H2AX or whatever. I mean, any repair protein, and then you see you can write the GSI using the microbeam. But here I want to show you a couple of data on the bystander effect. So what can you do with the microbeam? You can say, okay, I shoot 1 alpha particle, 2 alpha particles, 3 alpha particles in the nucleus. And then I ask the question, how many alpha particles I need to cause neoplastic transformation? Now, if you do that, this was an experiment done in New York at the Columbia University. You see, this is the Okay, in the points, the spheres here, the circles here, I'm sorry, are the average number of alpha particles per nucleus calculated with the Poisson statistics. And this is the transformation frequency. So you see that it goes up when you have 0, this is the control. Then you have 1, 2, 3, and so on. It goes, there's pretty much here, there's a sort of saturation here. But if you look at the exact number now done with the microbeam, you see that apparently 1 is not statistically different from 0. What 2 is already very close to what you would expect from the Poisson statistics. So in other words, you see, the transformation frequency per 10 to the 4 surviving cells, these are always the C3H murine cells. This is the control, this is the exactly 1 alpha particle, but if you do it with Poisson distributed, this is the control and this is the mean of 1 alpha particle. So apparently the microbeam unraveled that 1 alpha particle is not able to transform the cells. You need more than 1. This is very important in radiation protection, because in most cases, even for radon, you don't expect to have more than 1 traversal per cell. So you say, okay, but then we don't need to worry about radon because, you know, it's all done. No, because of the bystander effect. Now, you can have another scheme now where you irradiate this cell, but you don't irradiate this cell. So this is how it looks like when you have a detector that shows you, this is in the cell, this is out of the cell. If this is shown here, you see, the triangles are 100% microbeam irradiated cells. So the white triangles here. So you see 1 exact number, 1 alpha particle is very low and then goes up 2, 3, and so on. But now this black spheres is only 10% microbeam irradiated. So you only irradiate 10% of the cells in the field, okay? And look here, very surprisingly, if you want, pretty much the same here, but here you have more. So surprisingly enough, in this situation where you target this one, is likely that you can induce transformation in this one, the bystander effect. And also, surprisingly, which is shown here, if you target the cytoplasm, not the nucleus, you can find an increase in the number of mutations, nuclear mutations, coming from targeting the nucleus. Many particles. You need the high dose if you want the cytoplasm to induce mutations in the nucleus, but it's possible. And that's an experiment going straight to the transformation in vivo this time, in vivo. So what is this? This is a particular system for medulloblastoma, which is a brain tumor. It's called the PATCH1 radiosensitive mouse. This is a mouse which has a mutation in the PATCH1 gene that makes it very sensitive to medulloblastoma. Now, this mouse was irradiated either whole body with radiation, and you see it here. This is the medulloblastoma-free mice as a function of the time after irradiation. Now the black curve is the whole body irradiated mice as a function of the time. You see that at 3 gray, 70% of the animals get medulloblastoma. So it's very sensitive. It's a very sensitive system. This is now the control. You see, in control, you have no medulloblastoma. Now, what is the red curve and what is the black curve? The red curve is a mouse irradiated in this situation. So now you shield the sensitive part for medulloblastoma, the brain, and you only irradiate the body. And look, now you have an increase of medulloblastoma. You have many, not as many as here, but you have some 30%, 40% of the animals giving medulloblastoma. And if you ask the question, this is a question asked by the referee. I was a referee in this paper, so that's how it happened. I say, how do you know for sure that there is always some stray dose, some stray dose. Even if you use a shield, you have some stray dose to the brain. They measured that this stray dose was 0.036 gray, and then I asked them to irradiate it with 0.036 gray, and they did it, and you see that there is an effect, but it's definitely much smaller than the bystander effect. Or you see, bystander effect is more a term used for cells. This is really more like a tissue non-targeted effect. So you can have cancer in the brain irradiated in the belly. And this is really strange in a way, if you think about it, but clearly there is a role of the immune system, of the nerve system. We don't know exactly, but clearly this is the case. We don't know exactly because the pathways, this is an example of the possible pathways that you can have in the bystander response. And you see that some pathways are really related to cell-to-cell junction, but others are mediated by the immune system. This is a macrophage, and that mediates the immune response that can transfer the inflammatory response that comes from the cells that has been actually hit by radiation to other cells. The final point in the lecture is a little bit of modeling. We saw a lot of modeling of cell killing. We saw it a lot. What about modeling radiation-induced cancer risk? There is no doubt that is interesting. What I will discuss here is not so much the low dose part, that as I told you, we don't have many data, but more the high dose, because that's important for radiotherapy when you have normal tissue exposed to high dose. And also we will focus on leukemia because that's one of the main cancers that can be induced by radiation and is developed in short time after exposure. So if you want to make the model, I mean, you can start from this is a picture I showed already in biology, this multi-step approach. You have to simplify it a little bit. So you can imagine that you have initiation, then you have some kind of migration, promotion, and then you have cancer. So, but of course you also have inactivation from the cellular point of view, and you have repopulation. So the cells that are not dead will repopulate the tissue. And then of course the progression step, that is extremely important, which is probably more important than the initiation. So, okay, sorry. Inactivation, initiation, repopulation, this can be modeled. Generally, you model the excess relative risk. So the excess relative risk is the number of cancer cases in the irradiated population divided by the number of cancer cases in the irradiated population minus 1. So if there is no difference, if there is no effect of radiation, this should be 0, okay? If it is 1, for example, this means that this ratio is 2, so that you have 2 times the number of, it's 100% increase. You have 2 times the number of cases here than here. And you can write ERR in a model like the product of a constant, which is of course constant but depends on time, age, sex, but is independent on dose, and M, which is a pre-malignant stem cells after completion of repopulation. This depends on the dose. So the problem that you have is that these are the data for the lung cancer excess relative risk as a function of dose for Hodgkin's lymphoma data and for Hiroshima and Nagasaki. Now, Hiroshima and Nagasaki is necessarily related to a relatively low dose here because no one will survive 15 gray, 25 gray. But for Hodgkin lymphoma, which is partial body radiation, you can reach these doses. So if you take the data from Hiroshima and Nagasaki and you use a linear quadratic approximation, where you say, okay, the induction term is gamma d plus delta d squared. But then I have the cell killing, which is x minus alpha d minus beta d squared. So if I write this one as, induction normalized to the cell killing, the curve should be like that, which is not really nicely fitting the data. So what is missing here? You see the initiation, which is linear quadratic, the inactivation, which is exponential. What is missing is repopulation. So you have to take into account the fact that there is repopulation. So that the desire to keep constant the number of stem cells counteracts cell killing, but the proliferation of the malignant cells increase the risk. And in fact, you see what happens with the lung cancer excess relative risk data here. And for leukemia, this is also, it's even more complicated. You have to use a migration between the bone marrow compartments exchanged by blood. So the bone marrow is where the stem cells for the hematopoietic system is, but they can migrate, of course, they can move in the different bones. So if you write these equations, let's say for every bone marrow compartment and the blood, you know, you have the inactivation, the initiation, repopulation, migration, which depends on dose and active bone marrow in each compartment. You get a set of differential equations. And then you come out, you see this is now leukemia risk as a function of the mean marrow dose from fractionated radiotherapy and brachytherapy, that are two types of brachytherapy, and you see that the model is able to fit. So in conclusion, radiation is a mutagenic and carcinogenic event. There is no agent, there is no doubt about that, but is relatively weak compared to chemicals or viruses. In vitro system can provide quantitative information, but as you have seen, they are not really representative of the complex in vivo situation. Animal studies are more realistic, but as you have seen, they completely depends on which strain you use, which kind of model you use. Epidemiology is, of course, the key for radiation protection, but there are few data, and at low dose, the uncertainty is so high because of the low incidence that is difficult to expect that you will solve the problem. For many years, the mechanism was supposed to be multi-step with the involvement of the hits, let's say in the multi-steps were dictated by activation of oncogenes or inactivation of tumor suppressor genes, but these days, aging inflammation is really the new paradigm. And finally, heavy ion, we have seen the RB-LET is probably similar to the one for survivors. Animal data suggests the RB depends on the tumor. It's not depends. Leukemia is an RB. Hepatocellular carcinoma is a different RB. Epidemiology, again, if you take radon in the houses, is not solving the problem. So I hope this gave you an idea of the problem. Thank you very much, and we will discuss this lecture next time.